gives me great pleasure in introducing Dr. Chandrashekara, and he is one of the brainchild for this webinar series, and he is the Medical Director, Chandri Rheumatology and Immunology Center and Research, and he has a lot of affiliations, Vice President, Indian Rheumatology Association, President, Indian Rheumatology Association, Karnataka Chapter, General Secretary of the Society for Information Research, Managing Trustee of the IART, and then Executive Director, Editor of many Chandri journals, and also Faculty and Program Coordinator. He has several awards to his credit, the important one being the Sir C.V. Raman State Award for Young Scientists, Medical Sciences from the Government of Karnataka, and 2019, the Indian Medical Association Pune Annual NIO Oration Award, and many other awards. And he is an authority on autoimmune diseases. He has over 100 peer-reviewed publications to his credit. Many times actually, I've been in the medical field for last 30 years. When we talk about any forum kind of thing, we always address actually, clinicians and basic scientists kind of thing. But now, slowly actually the term actually physician scientist is picking up, and I consider actually Dr. Chandrashekara as a physician scientist. If you can look at his talk, actually you are going to actually see you cannot differentiate him as a doctor and a scientist kind of thing. He has actually a very blunted level of training. So he's going to talk on role of innate immune system in maintenance of inflammation, autoimmune diseases and drug targets, a way forward. Over to Dr. Chandrashekara.
Thank you, Dr. Subramaniam. Can I share my slides?
Yeah.
Yes, sir. Go ahead, sir.
Yeah, sure. Yeah, I think my slides is visible.
Yes, sir.
Yes.
Go ahead.
Yeah. So I think my previous speakers have made my task much more simpler. From the last 2 days, I think we have heard in detail about how neutrophil netosis can create trouble as a part of innate immune response. And today we heard about the macrophages and 2 of the NK cells, how they can play an active role both in autoimmune disease and sustaining inflammation. And also we have looked into how various receptors and various innate immune methods and processes can interfere in both the development of the disease as well as in the sustenance of inflammation. I think even to the date, what McIncoff said that we have yet to understand different dimensions of inflammations. I think we talk about inflammation. We know there is inflammation is both beneficial as well as harmful. When it is too much, it causes too many troubles. When it is too less, we don't get the job that is to be done. So that being the case, we need to work a lot on understanding the inflammation. So adding to the problem, we have another major questions. The major question we have is this inflammation, are these autoimmune disease? Who is responsible? I mean, it's always a problem in any democracy or anarchy when it happens. Who is the kingmaker? Who is responsible for everything? Who controls the system? Who is the reason for good and bad? Let's take the current scenario of the worsening of the second wave. We have a confusion. We point the fingers in the politicians and administration saying that they didn't bring in the rules and regulations and didn't control the crowd. That is responsible for the worst of the second wave. And parallelly, the administration blames the people that you didn't care about it and you didn't follow the COVID appropriate behavior. That is responsible for the second wave. Whatever it is, the second wave has struck all of us. has put all of us in the home, not able to generate, or has a heavy toll on the economics, etc. But the problem or the trouble is who is responsible for the trouble or the mess we are. And so also are the confusions that we have. Initially, we thought it is the adaptive immune response or adaptive immune system is responsible for the autoimmune disease. We always considered that the T cell or the B cell, which are autoreactive or recognize the self-peptide, may be the orchestrating or the master, which is responsible for the development of the disease. Slowly, we did understood that we have a lot of autoreactive or self-recognizing antigen or self-recognizing receptors or self-recognizing autoantibodies, what we call as natural occurring autoantibodies, naturally occurring repertoire, which is responsible to identify the self-antigens, which are present in both innate immune response or adaptive immune response. As we started realizing the confusion, who is the master or who is the beginner of this autoimmune disease or autoinflammation question remains still continuous. I think to look at one of the interesting paper in the evolutionary biology, where we could really see that there is a game that is continuing. So that whether it is the self-recognition system, which was originally present in innate immune system, has fathered the adaptive immune system as a consequence of this interrelationship, a sort of architectural constraint between both adaptive as well as the innate immune system is responsible for the development of autoimmune disease, especially promoted by excess of hygienic environment, reduced exposure to microbes and microorganisms, vaccinations and promotions of environment pollutions, etc. Are they all pushing either of the system or both the system together to keep a constant or a persisting inflammation, which over a period of time becomes autoimmune or autoimmune disease process? Whatever are the constraints, there are few issues which we need to remember. The role the innate immune system works with adaptive immune response. We all know from our knowledge on immunology and knowledge of working on immune responses, adaptive immune response need a constant support and a push from innate immune system. We know that the primary effector cells of the adaptive immune response are the recruitment of neutrophils, recruitment of NK cells, recruitment of monocyte macrophage system, and to greater extent some of the killing cells of course, the effector cells of the adaptive immune response do play a lot of role, but we have a lot of innate response cells being recruited for as effector cells. So though they are there, they do behave or they do adopt or they follow the commands from the adaptive immune response, they still act independent in certain of the points. And as we all know that TLRs don't recognize an antigen which is lying free. It need to be presented by an antigen presenting cells within few exceptional antigens. So also the B cells. B cells do needs they being presented by the antigen presenting cells. You need a constant continued antigen presentation by the innate response and also secondary signals which come from innate immune response for an adaptive immune response are the one which maintain or continues to stimulate or persist adaptive immune system response either on self or on non-self proteins. So thus I can say that initial response is guided by innate response, could have twists and turns based on varieties of signals which is received by the adaptive immune system by the innate immune system. So that makes the argument clear that probably the drive force for the variations, twist and turn could be by innate immune system. To argue it further, to know it further, we also know that innate immune response, I think yesterday, the first lecture as well as the mitosis and second lecture has clearly shown that the immune response, the system response, especially the innate immune responses to the damage-associated molecular patterns, DNA, and varieties of other degraded cell products. And also it's been known clearly that innate immune response can recognize and respond to the specific patterns. And also there are varieties of participating components which could interact with antigens and others. So in addition, the constant development or the adaptive immune system to get into a good response you need a constant and a continuous interactions. It's not that once the antigen recognition occurs by adaptive immune system, the innate response no longer has a have a requirement to present the antigens. It's not the case. The antigen presentation, antigen exposure to the adaptive immune response is continuously required from the innate immune system. So that being the case, there is a continued stimulus or a continued push, which is essential for adaptive immune system to go on in generating an immune response as well as the inflammation. So it's clear now the supporting facts are the proactive role of innate immune system is antigen presentations, triggering the inflammation by necrosis, apoptosis, or the other degraded product, and overall over or under expression of innate immune system component can certainly result in autoimmune disease. I think it's very clear for us from many studies that are published over a period of time, clearly mentions that these three key observations makes it a point that a continued trigger or a continued support or a continued push is required from innate immune system for the persistence of autoimmune inflammation, or for that matter, any inflammatory response that is generated either against self or non-self antigens. So in a nutshell, what I can put it as you could see from NK cell, B0 cells, macrophages, neutrophils, name the cells, they act, they start, and they sustain an adaptive immune response with specific TCR or epitopes and BCR receptors and antibodies. So there is a constant interaction that occurs between the innate immune response. Certainly it acts, starts, sustains the adaptive immune response for continued endeavor or continued persistence of inflammation. So how does it trigger? I think just take infection as one of the good best models. So we know now clearly from the last 5 discourse that any of the exposure to the antigens, they are presented through the varieties of receptors, influenced by the varieties of factor, including the genetic and other predisposition and other innate factors. They in turn activate many of these innate cells, which includes NK cells, dendritic cells, basophil, monocyte, macrophages, neutrophils, which in turn trigger the continued secretions of varieties of cytokines and varieties of presentations and the several number of cytokines, which in turn activate self-reactive or autoreactive or the auto-antibody productions, which in turn reinforce the process of inflammation. So thus you have a constant, the backward as well as forward flow of stimulus in continued maintenance of inflammation or autoimmune responses. So I think Dr. Giridhar has completely explained the dual role, how the natural killer cells in causing varieties of tissue injuries as well as protecting the tissue injuries. And this has resulted in, I think you have heard our previous 2 speakers clearly saying that there is a change, there is an adaptation, there is a differences which happens within the single cell, what we otherwise used to call as natural killer cell, otherwise we used to call as macrophages. And yesterday, I think in netosis, we have, in the neutrophil discussion, we said that the neutrophil has 2 different formats and changes its shape and functionality depending on the different requirement. And even Dr. Giridhar as well as Dr. Vani said that there is a memory or there is some amount of changes that the monocytes, macrophage, and NK cells alter themselves to the required circumstances. Now we started moving from, we always used to continuously say, the continuous classical teaching is innate immune response, don't have memory, adaptive immune response, memory. In 2006, when another paper that there is a possibility of memory within the innate immune response. But when we talk of memory, we always talk of them to be more precise. There should be some adaptability. There should be some changes which should keep that memory on. So probably the memory that innate system has, because being an old system, probably the memory that it can have was as limited to as our older computerized systems, vis-a-vis compared to the latest generation computer systems. Probably the possibility in which the innate immune response can manage its memory is either by undergoing a transient or a stable change within the expression of ligands and receptors, or altering the secondary and primary signal pathway, either overexpressing some of the factors which are responsible for a quick transmission of the signals, or underexpression of certain signals which could be otherwise inhibitory and could be deleterious for the possible suspected effect or possible suspected effector response that was required, or shifting in their phenotype from M1 to M2 macrophages. Thereby, they are expected to undergo a transient or a stable change, which we call it as possibly the memory or the change that would have been happening in the innate response to adapt to a necessary or adapt to a necessary circumstances. So slowly we started reworking on the terminology, because I think it's very difficult to call it as memory because it's not specific. The change that occurs either in the M1 to M2, or the changes that occurs in NK cells, or the changes that occur in neutrophil, etc., are not unique to the single antigen or a single protein that what we see in adaptive immune response. In adaptive immune response, all of us know that the TCR goes for a configurational changes to identify the epitope on the antigen. So also the immunoglobulins, so also the B cell receptors. But that is a very specific memory. It's a sort of an absolute memory. So that's why we slowly moved into use a terminology what is called as trained immunity, where immune system has been trained, modified, or adapted to a certain set circumstances. So slowly we also plan to expand the test to expanded trained immunity, because it's not just one or two cells which works, it works with multiple cells to have a unique requirement, or a requirement which is being threatened to the host. So based on that, the immune system, innate immune system, goes for a sequential changes or a change which is sustained for quite a bit of time, what we call as trained immunity, or which is otherwise was, since more elements are involved, they try to use a term called expanded trained immunity. This terminology was first introduced by Mihai Nitae. I think I'm sorry about pronouncing the name. He first time proposed a term called trained immunity, where it programs an innate immune response, both in health and disease. He proposed a few changes like epigenetic reprogramming in innate immune response, like histone modification, DNA methylation, modulation in miRNA, as well as in the long non-coding RNA sequences, which could adapt or change, make it more permanent to have a quicker adaptive state, wherein it could either induce mucosal tolerance, or it could limit the tissue damage by infections, or can go for a training program, especially against what we see with a non-specific protection by vaccines, that what we thought as BCG having some protection against COVID infection. Probably we were not right on that particular aspect, or some of the vaccination programs, which could give a sort of trained program, which can result in innate immune system maturation that could result in a good adaptive state. When it goes to a maladaptive state, which we see in sepsis, because of the severe inflammation, immune system goes for immune paralysis, where a sort of non-responsiveness, which can cause in a hypoinflammation and a susceptibility to infection, which we see post-sepsis. It's a maladaptive state. And similarly, an hyperinflammation state, which we see in most of the autoimmune disease, including the chronic inflammatory conditions of vessels, what we call as atherosclerosis. Here, most of the time, the innate immune response goes for a trained state or a trained status, wherein there is a continued persisting inflammation, which could result in an ongoing benefit if it happens in state of infection, to an deleterious effect if it happens in circumstances where it is not required. So during induction of a trained immunity, I think my previous speaker, Dr. Vani, has touched on that. There are a lot of transcriptional reprogramming occurs in the increased activations. There will be rewiring of the cellular metabolism with a shift towards glycolysis, mTOR pathways, glutaminolysis, cholesterol synthesis, and varieties of changes which occurs, which also, as I showed in the previous slide, the modulation of the function of the epigenetic enzymes and enables the increased gene transcriptions, augments or alter the expression of the surface receptors and cytokine expressions, also alter the secondary signals. By these adaptations, they make the innate immune response either vulnerable to hyperresponsive or potentially keep them active to hyperrespond to a given set of stimuli. So there are varieties of things. I think our previous speakers has touched on. There are varieties of changes that occurs within the macrophages. They're trying to reclassify and recluster them. We see them in a significant status in patients who are suffering from lupus. And similarly, NETOS, we know very clearly, I think my previous speaker has spoken at length, so I've got all these slides of NETOS. Finally, just to come to a conclusion, to draw the attention that in NETOS triggering or a persistingly causing autoimmune response, we now have a way to inhibit the NETOS using PAD4 blockers, which are of course a potential things which are getting explored. Others like colchicine, dapsone, etc., which can reduce the NETOS. Thereby, if NETOS is one of the constant trigger, which we are suspecting to be one of the part and parcel of the disease repeated proactivation in lupus, we can work it on to figure out how much the NETOS is contributing currently for the disease and propose or plan the inhibitors at this point of time. And this is how if we can understand and think about how an innate immune system is constantly hyperactive and triggering an autoinflammation, can work towards in managing or reducing the inflammatory status. Similarly, there are changes, as I suggested, in the surface receptors and ligands. I mean, there are many studies which are slowly showing that altered expression of TLR. For example, in non-alcoholic hepatitis, NASH, TLR4 expression has been shown to be increased. And similarly, in psoriasis, TLR7, an antimicrobial peptide of which is associated with self-derived DNA, is supposed to be the constant stimulus for 7 and 9, which could result in psoriasis. For example, they have demonstrated by using imiquimod, which is a potential stimulator of TLR7, can induce a psoriasis-like dermatitis. So what we meant here is the alteration or a change, an adoptive or a trained response that occurs in the innate immune system could be one another factor which we often don't consider when we are managing autoimmune disease, maybe the responsible factor which could keep the disease process active. And you could see that considering this, there are many newer molecules and many of the newer interventions are under clinical trials. And we know some of them can potentiate an immune response, especially when we are handling malignancies. Some of them can inhibit the hyperactive responses, can help in reducing the inflammatory process. So intervention at these levels can also help in altering, augmenting, or attenuating the responses with reference to the autoimmune or persisting inflammation. So we do now see that there are many studies with reference to autoimmune disease. I just take two examples, the common diseases like rheumatoid arthritis and SLE. In both of them, we see, for example, the circulating monocytes are hyperactive. There are higher ATP levels upon lipopolysaccharide stimulations. There is upregulated glycolysis, increased expression of mTOR. And we also see almost a similar changes even in lupus, but some are little different and unique to lupus. So there are certain changes which are unique to each of the disease, and some of the changes which are chronic to or common to both the diseases. So in a nutshell, I think as I could see, there are possible different mechanisms by which the innate immune system goes for a trained immune response or a sort of memorized or an altered immune responses, which could be either metabolic or it could be because of the constant changes in the antibodies, or it could be change in the epigenetic reprogramming, or because of excess of production of pre-inflammatory cytokines because of the reprogramming in that. So these can be done by interfering the metabolic pathways. I think There is work which is going on repurposing the metformin, repurposing the drug like rapamycin, which can work in mTOR pathways. We can use multiple antibodies to block the expression of certain of the cell surface receptors. And we can use some of the small molecules like JAK-STAT inhibitors and other inhibitors which can come in the way of reduction the expression of the secondary signal pathways. Varieties of nanoimmunotherapeutics programs are going on. I think when I was trying to search for the recent studies, I could see in the last 10 years, each year, more than 100 papers are published with reference to innate immune response as well as the autoimmune disease. That clearly suggests the interest that's going on across the globe with reference to the possible major role the innate immune system is playing with reference to these autoimmune disease and inflammations. Over in few years to come, we may have different methods from a simple repurposing of a drug to a complex TLR7 or TLR inhibitor drugs, which would be more useful in treating and managing these. So it clearly suggests that there is a lot of researchable avenues and places which are required with reference to innate immune system. So how should I handle that? Because now the trouble is, so far we were thinking that if I manipulate adoptive immune system, I can really make through with reference to autoimmune disease. It's clear that it's not just one system, you need to handle both the systems. I think that Giri showed in his slides that if I use a rituximab, an active NK cell is required to kill the B cell. Otherwise, the rituximab may not be much useful. So there is a participation of immune system even to help or assist in what the treatment we need to work with. So it's best to handle both systems. So looking as a single focus thing that probably if I manipulate adoptive immune system, I can do the job. Coming back to the second wave could have been largely prevented if both administration as well as citizens would have participated in a better manner. So what really happens is when a system goes for a task, probably both the systems are work together in either destabilizing or doing the things better. So that being the things, we always worried that what's the best way of intervening? Should I go stepwise? Step one, interfere in innate immune, then handle the adoptive immune, or handle adoptive immune and then come down to step one? Because the problem that we face is if I block both the system, what is the possibility of exposing the host or exposing the patient to a severe infections? So that's where we are worried. Shall I go with a double hit? If I freeze both the system, the person may be exposed to an increased infections. So either we could go with a stepwise or we can go with a double hit, which are always of a concern, or a balanced approach, step by step, try to work on and try to alter argument such that you can first block the second signal, which could reactivate or reignite the autoimmune processes, and a balanced or a target specific protocol based approach. When it comes in place, probably we would be more effective in managing autoimmune diseases with least possible adverse event. Thank you very much for the patience hearing.
Thank you, Dr. Chandrashekara, for your memorable lecture on innate immune memory and delineating the possible mechanisms and drug target. Once again, you proved as a very good teacher, doctor with doctorate qualities. Thank you.
Questions? Dr. Balasubramaniam, questions?
Yeah. Please. The forum is actually open to questions.
Yes.
Yeah.
This is Padma Shastri. Padma here. And as Dr. Balasubramaniam just said, Dr. Chandrashekara has been a fantastic teacher. For somebody who would not have understood these things in a very complex manner, he has very simplified it. Can you hear me?
Yeah. Thank you. Thanks, ma'am. Yeah, please.
Now the question is, with innate responses, with autoimmune diseases, inflammation all together working, what is the prevalence and the clinical outcome in COVID patients with autoimmune diseases?
Shastri, we could not hear you.
Okay. Yeah. Should I repeat? Yeah.
Please.
With innate responses, autoimmune diseases, inflammation, all working in together. What is the prevalence and the clinical outcome of COVID-19 infections in autoimmune disease patients?
Yeah, I think we are shortly publishing the experience from India. I think we just made our manuscript ready. What we did find that there was no significant difference with reference to the outcome of the patients having a COVID infections. What we saw that their need for oxygen, need for ICU, and the overall mortality was not very much different from what it was in the general population. Even the risk of a ICU or the risk of oxygen requirement was more or less similar to what was there in the general population. But this was more particular to the patients whose autoimmune disease was under reasonable control. But the patients with an active, proactive disease, probably they had real trouble in worsening of their autoimmune disease along with COVID. They had to manage both the conditions little bit of difficulty and having immunosuppression at one place and also having anti-infective disease. In that case, there was a moderate increase in the morbidity and mortality. I mean, it's not that there was a very significant increase in mortality and morbidity. But what we have observed of late is some of our patients, I think we have now, I think in the second wave, I had in the video consultation, I could figure out at least 6, 7 of the families. Most of the families, 5 members have been infected. The patients with an autoimmune disease were on hydroxychloroquine or with a moderate immunosuppression with a good disease control. They had a milder disease compared to their other family members of the same age. So probably this immune modulation or immune suppressants which are given do have a protective role in rolling into the cytokine storm, because I think you now know that we are now utilizing half of the rheumatology drugs into the management of the cytokine storm. We are now using JAK inhibitor, we are using anti-IL-6 drugs. Hydroxychloroquine in the very beginning has made a lot of controversy, which is a drug which is used and has clearly shows that a probable control on the immune modulation can safeguard these patients from going into the cytokine storm.
Just got one question. In cancer therapy nowadays, is more and more using the target therapy. In the immunological innate system, the target therapy is now how common it is and how it is mechanism of the target therapy.
Sir, I think they are potentially using the TLR7 activators. Some of them are phase 2 clinical trials, which can enhance their immune response. Probably, I think Dr. Girish will be able to tell more about the NK cell and CAR T cells, which are the other areas where they are using the innate immune response. And I think Dr. Vani should also add that because macrophages are also being targeted to tune and train and put it for anti-cancer therapies.
So like very nice question that how we can exploit these innate cells. So we know that adaptive immune cells are anti-toxicity, and that's process related. But antibody doesn't require to have any antibody against any target. It will just go and bind. So what people are using is mixing these two functions. That one, which is the receptor which interacts directly, take that receptor, put into the cell which has the property of spontaneous lysing killing the target, like NK cell. So in NK cells, you put the antibody fragment, but receptor in the intracellular receptor will be part of NK cell. So the surface recognizes just like antibody, but signal goes in the NK cell just like NK cell receptor. Now NK cells are tuned now. The moment receptor recognize to kill any target. So now you have any specific or designed or the primary the NK cells which can specifically recognize your specific target and only kill those target which you have intended. The only drawback is that when it kills, it's not just the killing process. It also has a lot of cytokine storm. And that is where one of the major limitations with most of these chimeric T cell or chimeric NK cell adaptive transporter. But you have specificity, but you also end up with the cytokine storm, which build up when the cells get activated simultaneously in other cells. Yeah.
Thank you.
Hello. I have one more question. Can I? May I?
Sure, please.
Hello.
Please go ahead.
I have one more question, Dr. Chandrasekhar.
Please.
Yeah, please.
Please, Dr. Shastri,
Hello?
please you can. Yeah, please, you can ask your question. Dr. Padma?
I think
Hello?
it's a network issue or something.
I
I
think
think
there is
she
some
has a
network
network
issue.
issue. Yeah.
Yeah.
Any other questions from any of the doctors and attendees who are there?
Dr. Chandrasekhar, I have a question.
Yeah.
This is regarding the usage of intravenous immunoglobulins for management of autoimmune diseases. It's a very nice perspective with respect to innate immune components participating in the trigger of autoimmune diseases. So would you think the intravenous immunoglobulins that are already used in therapy might be working also on the macrophage compartments with respect to the innate components you spoke about?
Yeah, I think, yes, I fully agree with that. Yes, these intravenous immunoglobulin do interfere even in the macrophage functioning. I think we had a real good discussion on this IV immunoglobulins and macrophage activity in the last of our series in the April-May. I think definitely these immunoglobulin concentrate has both the natural occurring autoantibodies and naturally modifiable antibodies, which could modify the functionality of the macrophages as well as the neutrophils and probably the other component, all other components. That's how their efficacy, that's how they are thought to be functioning. But I mean, truly speaking, we really do not know how exactly these IV immunoglobulins help in these circumstances. One of the hypotheses is macrophages too.
Right. I mean, Dr. Srini's lab works on that, of course. That's the reason that I asked the question. But we do know the regulatory functions with respect to disease and macrophages. I was just wondering with respect to the, the Leuconeal's, proposed hypothesis with respect to epigenetic changes, etc. Has anything been shown with respect to IVIG modulating any of that? And I mean, a direct cause effect of an etiology of autoimmune. Via innate.
Right. And I mean, I'm not sure because I mean, I didn't see in that perspective any publications. I'm not really sure about that.
So
Thank you.
I think Dr. Srini's lab has published that information to modify macrophages, including the regulatory T cells participating with the IVIG.
Right, Girdhari. But the thing is, the question was with respect to the specific components of the innate immune components contributing to the autoimmune diseases. No, with respect to etiological, with respect to adaptive immune components and the etiological agents, such as release of sequestered antigens or molecular mimicry, we have kind of pinned down some levels of etiological agents for the origin of autoreactive T cells or the autoantibodies. So, like that, with respect to innate immune components, do we have any such pinned down, associations?
I think,
and IVIG.
There is only one study where they have tried to use IgM against anti-dsDNA. The close are similar to the natural occurring autoantibody against dsDNA. There they have shown that it can reduce the intensity of nephritis as well as the lupus disease activities. That probably gives a clue that probably the IVIG concentration may have some of these natural occurring autoantibodies, which may be blocking the development of pathogenic autoantibodies. That's hypothetical based on the other study of anti-dsDNA IgM.
Okay. Thank you, Dr.
Thank you.
Any other questions? Dr. Shastri, is she able to connect? No.
No, I think she got disconnected.
That was a wonderful session. Dr. Vani, Dr. Giridhari, and Dr. Chandrashekara. It was very interesting, very informative Sunday, I should say. I think probably Dr. Biswas would like to say what is the next thing that we'd
I
like
think
to
is
do.
the good Sunday treat on immunology. And we have got stalwarts talking about the various NK cells, then macrophages, and then innate immunity and the target therapy. I think is we look forward for the next session will be on the June, July, isn't it, Dr. Chandrasekhar?
Yes, sir. Yes, sir. Yes, sir.
Yeah, and we invite a lot of our listeners that if they're interested, they can join this society. This is we have got a website. You can download the application form. We're also bringing out a book on inflammation, which is in the process and will be soon coming out. So it's a very good Sunday evening, and we applaud all the speakers and the chairpersons for organizing, for conducting this meeting. So have a good evening. Thank you very much.
Thank you.
We conclude
Thank
here.
you.
Thank you. Thank you, sir.
Thank you. Thank you.
Very nice session. Nice talks.
Thank Mr. Vasant and Mr. Anand for the back team support.
Welcome, sir. Welcome.
They have done a good job.
Exactly. Nice job, I think.
Welcome,
Thanks, sir.
Thank you, and take care of yourself.
Micro lab.
Right. Thank you. Take care.
Thank you.